All right, everyone. Thank you for joining us today. Today at Precisionary Instruments, we are excited to host Dr. Natanael Zarco for his webinar talk. So Dr. Zarco is going to speak about cell motility in glioblastoma. He is a research associate at Mayo Clinic in Jacksonville, Florida, where he's enjoying probably nicer weather than many of us on this webinar today. He specializes in brain tumor studies, particularly glioblastoma or GBM biology. He has a strong academic background where he holds a Bachelor's of Science in Chemical Pharmaceutical Biology from Mexico, and he earned his Master's of Science and Ph.D. in Cellular and Molecular Neurobiology in Mexico City. So currently, Dr. Zarco's research is focused on exploring the interactions of glioblastoma with the subventricular zone. And it's a very significant and important neurogenic niche that houses neurostem cells. His study dives into exploring how glioblastoma stem cells invade the subventricular zone and other brain regions. Hence, he will be talking about cell motility in this tumor. So with that, Dr. Zarco, I want to hand it over to you. And we are very thrilled to have you today for this webinar with our company.
Thank you for the kind introduction, Ivy. And thank you all for being here today. I'm going to start the talk. As Ivy said, this talk is going to be entitled Cell Motility in Glioblastoma. I am a research associate in Mayo Clinic in the Department of Cancer Biology here in Florida, where the weather is very, 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 very hot today. Okay, this is going to be the outline of this talk. First of all, I'm going to give you a brief introduction about the brain tumors, and with a particular focus on glioblastoma. After that, I'm gonna give you some aspects about the glioblastoma and the lateral ventricles, the importance of the cell migration in cancer progression and prognosis. What about the cell motility and the role of the chemotaxis in the cell migration? And finally, I'm gonna explain some of the experimental models we are using to study the cancer cell migration, and then we are gonna have question and answer session, right? Then the cell motility in glioblastoma, a brief introduction is these three persons, they have something in common. It's not that all of them were politicians. It's because all of them passed away about a very, very dangerous disease, which is a brain tumor called glioblastoma. The cell cycle is a very, very, very well conserved process in the cells in different species, in which the cells divide and produce daughter cells. They eventually can pass to different phases of the cell cycle, called G1, S, and G2, but eventually some of them can go or enter to the G0 phase, which is in a kind of quiescent state in which the cells are not proliferating, but they can come back to this state and then re-enter to the cell cycle and proliferate again. Well, in cancer, this kind of break it doesn't exist. The cancer cells proliferate and then is passing every time during the cell cycle, and then is producing several daughter cells. Here, the brain tumors is a proliferation of abnormal brain cells, which interfere with how the brain and the body functions. In most of the cases, requires surgical intervention. They can vary in size, location, and severity. Currently, there are like approximately 7,000 new cases diagnosticated every year only in USA. There are about 700,000 people only in USA living with a brain tumor, and from them, 130,000 people are living with malignant tumors right now. The glioblastoma is the most common and aggressive primary brain tumors in adults. Represent about the 50% of all the malignant brain tumors, and every year about 18,000 new cases are diagnosticated only in USA per year. Despite the standard of care, which includes surgery, radiation, chemotherapy, the survival is about only 60 months after the diagnosis. that means about 5% of the people that were diagnosticated with glioblastoma is going to survive for more than 5 years. But 95% of all of them are going to die within 5 years after the diagnosis. This is because the recurrence of the tumor is close to the 100%, and principally due to the treatment resistance and tumor invasiveness. Well, now we are going to analyze or explore the glioblastoma and the relationship with the lateral ventricles. And we are going to highlight the importance of the cell migration in cancer progression and prognosis, and its cell motility and chemotaxis. The glioblastoma location is important for the prognosis. Here in this figure, we can see two different kind of glioblastoma. In this MRI image, we can see some tumors that are close or are touching the lateral ventricles, and some of them are not touching or close to the lateral ventricles. The ones that are touching the lateral ventricles, they tend to recur at distant locations. These tumors that are contacting the lateral ventricles represent about the 50% of all the tumors, all the brain tumors. The patients that suffer these kind of lateral ventricles GBMs present a worse survival and a worse prognosis, as you can see here in this figure. So something is going there. Probably the GBM interaction with the lateral ventricle components is making something. So we explore that hypothesis, and then we first of all analyze the structure of the lateral ventricles. Here, the lateral ventricles, they compresses the subventricular zone, and also in the lateral ventricles we can find the cerebrospinal fluid, the CSF. In the subventricular zone, or ventricular subventricular zone, we can find a special population of cells called neural stem cells. The ventricular subventricular zone is the largest neurogenic niche in the adult mammalian brain. These cells, the neural stem cells, and they have a very special characteristics. They have a cell renewal, differentiation. They can differentiate to different cell types, and also they have an asymmetric division. In our laboratory, we were able to establish this model of the study, the interaction between the glioblastoma and the lateral ventricles, in which we injected or implanted the GBM cells, human GBM cells in mice brains, distal to the lateral ventricles and close to the lateral ventricles. As you can see here, the proximal GBMs to the lateral ventricles, they have a more immunoreactivity to Ki67, which is a marker of cell proliferation compared to the distal. And also the GBM that are distal to the lateral ventricles, they have a better survival curve. So another work in our laboratory, we also describe the differences in the structure of the ependymal cell barrier between these two kind of tumors, the proximal and the distal. In which we were able to see the penetration of GBM cells through the ependymal cell barrier and disrupting this barrier and touching the cerebrospinal fluid, which is in the lateral ventricles. After that, we decided to see what are the effects of this cerebrospinal fluid from different patients. In here, you can see here that the CSF from cancer patients, they promote the migration or increase the migration of GBM cells compared to the non-cancer CSF and the control. And also increases the percentage of the cell growth in the cancer CSF compared to the other experimental groups or the controls. After that, we evaluated the differential expression of by RNA sequencing to see which are the different genes that overexpress in when the cells are under the effects of the cancer CSF. And then we found different genes and different elements, and are modifying the expression. One of them is the SERPINA3 and the CD44. And also we decided to evaluate the different proteins that contains these different cancer CSF from different grades of gliomas, from glioma type 2, glioma type 3, and glioma 4, which is the glioblastoma. Also, we found the different proteins that are overexpressed in these samples. And then we focused the attention on one of them, the SERPINA3, which is also called alpha-1 anti-chymotrypsin. And we found that this had a very good contribution of the stem cell characteristics and enhance the tumorigenicity in glioblastoma. By different experimental approaches, we evaluated all these characteristics. Well, going a little bit back about the ventricular zone and the relationship with the glioblastoma, we said that in the subventricular zone, we found there is a special population of cells called neural stem cells. These neural stem cells produce neuroblasts, and then these neuroblasts, via chemotaxis processes, migrate towards the olfactory bulb. In this migration is directed by chemorepellent factors present in the CSF. One of these factors is the SLIT2. This SLIT2 protein acts in a gradient of concentration to direct the migration, interacting with its roboreceptor to regulate the neuroblast cell migration towards the olfactory bulb. Well, here we found the problem, the hypothesis. The problem is that the lateral ventricles proximal GBMs tend to recur at distant locations and have a worse prognosis. So the question was, can GBM cells respond to slit robosignaling in the same way as neuroblasts? And here we are proposing that this signaling pathway, the slit robosignaling pathway, plays a very good important role directing the invasion by chemorepulsion in other areas of the brain parenchyma in GBM. So first of all, we work with patients' samples, tissue, and we found we analyzed the expression of the different components of the signaling pathway. In this case, we analyzed the expression of ROW1 by mRNA and Western blot, detecting the expression of the protein, and we found that there is a significant increase in the expression levels of ROW1 compared to the low-grade gliomas and the non-cancer samples from patients. This was corroborated by TCGA databases, in which we found the same significant differences. Here also we found that the survival of there is a significant difference between the low levels of expression of ROW1 compared to the high expression of ROW1 in the survival prognosis of patients with glioblastoma. After that, we evaluated the presence of SLIT2 proteins in the CSF, and we found that there is an overexpression of SLIT2 in CSF from high-grade gliomas compared to the non-control and low-grade gliomas. Well, once we corroborate the expression of these components of this signaling pathway, we decided to evaluate the presence of these components in our study model, which is going to be cells. In this case, we work with patient-derived GBM cells, and here we are obtaining these cells directly from the patient from the OR, and then in the laboratory we were able to culture these cells. These cells are very well characterized in our laboratory by different assays: the glioma sphere formation, the pluripotent differentiation, and principally the tumor initiation by the injection of these cells in mouse brain and then the formation of the tumor. Here, we decided to analyze then the expression of these components now in glioblastoma cells. Here, we found that there is also an overexpression of ROBO1 mRNA levels comparing to the non-cancer cells. And also we found an overexpression of ROBO1 at the protein level. Here, we found a downregulation of the SLEED2 mRNA in these cancer cell lines, in the GBM cell lines at a mRNA level. So after that, we decided to evaluate the role of this signaling pathway in modulating the glioma cell motility. To do that, we evaluate the migration of these cells in 2D migration, and then a time lapse. In here, you can see the variation in the speed, where the cells are present, are in the absence of a slit, and then in the presence of a slit, there is an increase in the speed variation. You can see here a significant increase in SLEED2 when we added the SLEED2 to the media, increases the speed of these cells during the migration. And then there is also an increase in the total distance. We evaluate this in 2 different GBM cell lines. But we wanted to evaluate the chemotaxis effects of this SLEED2 in these GBM cells. And to do that, we used the transwell migration assay, in which we added the cells in the upper chamber, and then in the bottom chamber, we added the media plus FDS to promote the migration, to attract the cells to migrate through the membrane, which is here, and then we added the SLEED2 to the bottom chamber. Doing that, we are going to evaluate the chemorepellent effects of SLEED2 by impeding the migration of GBM cells towards to the bottom chamber. Here you can see here the results, in which we found a significant decrease in the migration of these GBM cells in the presence of SLEED2 in the bottom chamber. So that means the SLEED2 is chemorepelling the migration of these GBM cells. But we also utilized a chemotaxis chamber, in which we can establish a very good gradient of concentration of this SLEED2. Here we evaluated the migration towards to the lower concentration or the higher concentration of the chemorepellent. Here you can see here in both GBM cells, they were migrating in an opposite way of the concentration of SLEED2. Here also we can find that there is significant differences in the forward migration index in the axis of the X, in the X-axis. So that means this SLEED2 plays a very good important role in the chemorepellent effects of these GBM cells. So to evaluate in a deeper manner this signaling pathway, we had 2 different approaches, experimental approaches. First of all, it was the targeting of the expression of the receptor, in this case is ROBO1, or the targeting the interaction of the ligand, SLEED2. In the first one, we propose that the decrease in the expression of ROBO1 receptor can inhibit the chemo-repulsion of these GBM cells, and then inhibit the invasion of these GBM cells towards to the other areas of the brain parenchyma. So to do that, we utilize the same patient-derived GBM cells, and then we use the short heparin RNAs to knock down the expression of this ROBO1 receptor, and then we evaluate the response of these GBM cells in the presence or absence of SLEED2 in the culture media. Here is the corroboration of the knockdowns in both GBM cells. Now here, we use the same 2D migration assay to evaluate the different components of the cell migration. We found that in the two different knockdown experimental groups, significant decrease comparing to the control groups, the wild type, the anti-vector, and the scramble. In the total distance and also in the distance from the origin, the migration here in both GBM cell lines. And also using the transwell migration assay, we found that in the control groups, there is a response of GBM cells to the chemorepellent effects, obviously too in the culture media, but not in the knockdown groups in which these cells, they don't respond to the SLE2 in the culture media in both cell lines. So here we can conclude that the ROBO1 receptor is essential for SLE2 response in the patient-derived GBM cells. But then we were wondering if we can replicate these results in vivo. So to do that, we injected the different GBM cell lines, including the different experimental groups. And then we were monitoring the tumor growth by different assays. And here by luminescence, we were detecting the tumor growth. And then at the end of the study, we collected the brains and then made the histology to see and then to detect the different GBM cells in the mouse brain. So here you can see the three different experimental groups: the anti-vector, the knockdown 1, and the knockdown 2. And here you can see the immunohistology to detect the human nuclei, which is going to identify only human cells. And here also you can see the immunoreactivity for Ki67. This antibody only detects the Ki67 from human cells. So we are seeing here only human cells in this immunochemistry. So as you can see here, we can detect these cells, and then we evaluate the different invasion of these cells in other areas of the brain parenchyma. And there is a significant decrease in the migration of these GBM cells in the knockdown groups comparing to the empty vector, the control group, which is the empty vector, in both in the invasion and also in the proliferation. So here we can say that the low levels of RO1 reduces the tumor progression and increase the survival. Here you can see also the tumor growth during different during this period of time, and we can see a huge difference between the knockdown groups comparing to the empty vector, in which we can see a bigger tumor. This is also represented by a higher bioluminescence activity. Also, we evaluate the tumor volume in different slides of the mouse brain, and then we saw a significant decrease in the tumor volume in the two different knockdown groups compared to the empty vector. And also we saw that there is a significant increase in the survival in the absence of the low levels of ROBO1. Okay? So ROBO1 is participating in regulating all this tumor progression and the cancer cell survival. So here we were interested to analyze what are the molecular mechanisms of these effects. So first of all, we detect some morphological characteristic differences between the scramble and one of the control groups, and then the two different knockdowns. They have two different morphology. And then to analyze in a better way these changes in the morphology, we use the LifeAct GFP tool to label the actin cytoskeleton, and then that is going to allow us to study in a better way the morphology of the cells. So doing that, we first of all we're wondering if there is structural differences of organization in the actin cytoskeleton. And here you can see the different experimental groups, the scrambled and the two different knockdowns. And we found a significant difference in the orientation of these fibers, actin fibers. And we also found an increase in the number of fibers in the knockdown groups, surprisingly. But these fibers are shorter compared to the control group, to the scramble. And then they are also less in the thickness of this of the fiber. So it looks like the ROBO1 modulates the actin cytoskeleton structure by regulating the number of fibers, and then the length of the fibers, and then the width of these fibers. We also were interested to analyze if there is some differences in the actin cytoskeleton dynamics. And to evaluate that, we use the FRAP technique, which is the fluorescence recovery after the photobleaching. In here, shorter times of recovering of this fluorescence represents that there is a more dynamic actin cytoskeleton. So this is more related to a more pro-migratory phenotype. If this time of recovery takes more time, that is more related to this less dynamic actin cytoskeleton, and then this is more related to a less pro-migratory phenotype. So in this case, we evaluate this FRAP, this fluorescence recovery in the different experimental groups. Here you can see first of all that we found significant differences only in the knockdowns and the scramble control with any kind of extra experimental approach. But in here, in these two last panels, we can see here that in the presence of a SLE2, there is a significant decrease in this time of recovery. So this is more related to a more pro-migratory phenotype. This happened in all the three experimental control groups, but not in the knockdowns groups, in which there is an increase in this time of recovery. So that is more related to a less pro-migratory phenotype. And also there is not a significant difference between the presence or the absence of SLE2 in the control in the culture media. So the cells can respond to SLE2 because the absence or the low levels of ROBO1, which is the receptor. So that happened also in another GBM cell line. So ROBO1 is regulating this dynamic of the actin cytoskeleton in GBM cells. So then we were interested to analyze the expression of actin associated proteins. In here, we were analyzing the phosphorylation of two very important proteins related to the actin cytoskeleton, which is the cophylline. Here in this panel, we found an increase in the phosphorylation of cophylline. This phosphorylation of cophylline keeps the cophylline in an inactive state. So this is more related to a less pro-migratory phenotype. So we found this increase in the phosphorylation of cophylline in the two different knockdown groups compared to the control. And also this is also replicated in the presence of a SLE. But even if there is a slightly increase in the knockdown groups, it doesn't recover the same levels as the control groups. So in this case, we analyzed the phosphorylation also of ERM, which is another important protein related to the actin cytoskeleton. And in here, the phosphorylation of ERM keeps ERM in an activated state. So here we can see a decrease in the phosphorylation of ERM the knockdown groups comparing to the control groups, and also that happened in the presence of a SLIT2. So SLIT2 RO1 signaling modulates the activation of actin associated proteins. So then we were wondering if there is any differences in the surface and cell volume. And in here, using the same approach, the LIFACT tool, we were able to take different C-stack projection of the cells, and then we made a 3D reconstruction of these cells, which allows to study the cell volume and the cell surface area. So doing that, we were able to have this data and then analyze the changes in these parameters. So here you can see a significant decrease in the ratio surface volume in the knockdown groups comparing to the three different control groups, the wild type, the anti-vector, and the scramble. And after that, we evaluated the differences in the expression levels of different genes that are regulating or participating in the regulation of the cell volume in the cells. So here in all of them, we found an overexpression of these genes, the aquaporin 4, the NKCC1, the KCC1, and the NCC, in the knockdown groups comparing to the control groups in these cell lines. So that means that RO1 is also regulating or participating, modulating the shape of the cell, not only regulating the migration or related to the actin cytoskeleton, but also regulating the cell volume and the cell surface area in these GBM cells. So after that, we wanted to corroborate this finding and then we were restoring the expression of RO1 in the knockdown cells to see if there is a kind of rescue in these GBM cells, if they can recover the cell motility characteristics similar to the wild type. And in here we engineered an lentiviral vector to overexpress the ROBO1, and then here we overexpress this ROBO1 in the knockdown cells, and then we studied the migration of these cells in control condition and then in the SLIT2 conditions. We were able to analyze this data, and let me, I don't know why it's not reproducing the video. Maybe the compatibility between the Mac and Windows, I don't know. But anyways, the important thing here is that in the presence of SLIT2, the cells now, the knockdown cells overexpressing the receptor, the RO1 receptor, they are able to respond to the SLIT2. And now they can increase the speed of the migration, and also they can migrate more distance compared to the control condition in which there is no SLIT in the culture media. There is also a significant increase in the forward migration index in the x-axis. Well, the other approach, it was to targeting the interaction with the ligand, which is SLIT2. And in here, we were proposing that if we can inhibit the interaction of the ligand with its receptor, there is not going to be this chemorepulsion, and then the invasion in other areas of the brain parenchyma is going to decrease. So to do that, we analyzed the structure of these two different proteins, and then we determined the minimal components or minimal region of interaction between these two proteins. We then subclone this sequence, and then we produce the recombinant protein, and then we use that recombinant protein to has a decoy receptor to inhibit this interaction between these two, the receptor and the ligand. As you can see here, this is the experimental approach. In here, we can see here that there is a response in in GBM cells in the presence of a slit, but not in the presence of this decoy receptor in both cell lines, the GBM612 and the GBM965, and also in the cell migration here. This is a transwell migration assay. We can see here the chemorepellent effects of SLE2 in the GBM612 cells, but in the presence of these two different decoy receptors, there is a significant decrease in the effects of the chemorepulsion of SLE2 in both GBM cell lines. So that means also if we regulate the interaction of this ligand with each receptor, is going to modulate the migration of GBM cells. Well, the last but not least is the experimental models to study cancer cell migration. And here we are developing these models. Here we were also using the compresome from Precisionary Instruments, which is a very good device. In the past, we were trying to get a very good slices with any kind of distortion in the structure of the tissue, because the old device we were using, we couldn't get a very good slices from that device. So we found the compresome here, and then we used it to produce this beautiful slice. We also published a paper using this compresome in the literature, and how we can get these kind of slices and then study the cell migration in this context, in the tissue context. It's very important for us to study this migration directly in this context, because it has different, sorry. Because we need to analyze in a better context, in a more physiological context, the migration of these cancer cells. So in this case, we are not only using the mouse brain, but we are also using human tissue from the OR during the surgery, the remotion of the surgery of the tumor. Sorry, during the surgery, the neurosurgeon is getting these tumors. I'm so sorry. Okay, so in this case, it is very important to analyze the migration of these GBM cells in the correct context. So in this case, we are using also the human tissue from the OR. The neurosurgeon is getting this tissue, and then in the laboratory we are getting these slices and then evaluating the migration. So this is also a video in which we are implanted these GBM cells. So now we can evaluate the human GBM cells migrating in human brain. So I don't know why it is not playing the video here, but at the end we can evaluate the different parameters and different speed and different invasion of the GBM cells inside of the tumor, of the brain tissue, sorry. So in summary, we can see after this talk that the importance of the GBMs that are touching the lateral ventricles results in a worse prognosis for survival. And also there are factors present in the CSF that modulate the GBM characteristics. Also that the slit robot signaling pathway can contribute to glioma cell morphology, cell migration, and invasiveness. And this signaling pathway is also a druggable target, and we can see the need for develop new experimental models to study the cancer cell migration. So I want to give you many thanks to the Neurogenesis and Brain Tumors Laboratory here in Mayo Clinic, the Brain Tumor Stem Cells Research Laboratory, the different fundings from different federal sources and also non-federal sources, and principally the patients who can able to do this, to develop this research in our laboratory. So thank you so much, and then I ready to take the questions.
Wonderful. Well, thank you so much, Dr. Zarco, and please do follow Dr. Zarco or contact him. I want to open up the floor now to see if anybody had any questions. Please feel free to enter your questions or comments into the chat if you want me to read it out, or you can unmute yourself and also show yourself on video. I wanted to open the floor to everybody. Does anybody have any questions or comments?
Yes, I have one question.
Yes, please do.
Several questions, actually.
Go ahead.
Hello, Natanael. I'm Baruch.
Hello.
I don't know, you remember me. You lectured me several years ago at CINVESTAV.
Yeah, I think so. Yeah, I recognize your name, but I don't remember very well your face.
It's nice to see you're doing good.
Thank you.
Anyway, my questions are the following. So is there any isoform of this ROBO1 receptor that could take over the functions of this specific receptor?
There is another ROBO1 and another ROBO elements in this family. There is the ROBO1, 2, 3, and 4. In human brain or in brain, there is only expresses the 1, 2, and 3, and they can also have different function between the ligand. They can interact with the SLE2, SLE1, or SLE3 also. So doing that, they can also promote the migration of these GBM cells. But the most important one is ROBO1 and SLE2. I don't know.
Okay. And then, because this is like an important therapeutic target, what would be the therapeutic approach per se, like in the future, gene therapy, pharmacologic therapy, or what kind of approach?
Principally, I think pharmacologically. The goal of this research is to develop small molecules that can interfere with the interaction of this ligand and receptor, and then inhibits the migration of the GBM cells in a specific way. So principally pharmacological.
Yeah, that's important, because my main question here is, because ROBO1 is also involved in axon guidance and spine formation and everything that is like essential for plasticity in the brain. So is that going to be impaired as well if you use some antagonist of ROBO1, or what would be your thinking here?
Yeah, that's a very good question. Always the principal problem of all the therapy in cancer is the specificity. So in here we are proposing that the SLE-ROBO signaling pathway is one of the main ways to promote the migration of these GBM cells. So even if this signaling pathway is also acting in different contexts within the brain or within the brain, different GBM cells in the brain, uses this signaling pathway to migrate or to do the different process that they are participating. We can or we are proposing that inhibit this signaling pathway can provide an benefit of the patient. It's also previsible that there is going to be affecting another cells that they are not the cancer cells per se. But since these GBM cell lines are using more the signaling pathway, so we can inhibit in a better way the migration of these cells. Yeah.
Oh, thanks so much.
No, thank you. Thank you for asking, and thank you for being here.
Nice to see
Thank you
you.
for your questions.
All right.
And Dr. Sharma looks like he unmuted. Please help. Please go ahead.
Yeah, hi. I had a few questions as well, if that's all right. So my first one is I'm actually looking, hopefully doing PhD soon in glioblastoma. I'm looking a little bit more into the tumor microenvironment though. And I want to know, I can see you've used primary cell lines derived from patients. Did you use any sort of commercially available cell lines at all?
No, not yet. Here in our laboratory, we are using only primary cultures because it's one of the focus of the study. We have the different commercial cell lines like U87 or U373 or this kind of human GBM cell lines, but principally our investigation, our research is using the primary cultures.
Okay, brilliant, thank you. And then also, there was a paper a few years ago, I think, I remember, looking into macrophages and microglia and how they sort of stimulate co-invasion within glioblastomas. Have you looked into that at all, or is that something on your radar?
Not yet. We are planning to see that in the future.
Yeah, just thought that'd be interesting because it kind of tied in really well with what you've done. Brilliant talk, by the way. But yeah, just thought it tied in really well. So would be interested to see what effect that had in yours.
Yeah, thank you so much.
Thank you.
Oh my gosh. Well, thank you, everybody. And thank you especially to our speaker, Dr. Zarco. I hope everyone had a good time in the webinar tonight, and I wish everyone a wonderful week.
Thank you so much, Ivy.
Thanks very much.